Welcome back to the Cell and Molecular Biology course. I'm your course instructor NEET. In this video presentation, we will see the vesicle formation and transfer in cells. In the next few minutes, we will explore how cells create and move vesicles, which are tiny yet essential structures that play a key role in transporting materials within and outside the cell. Cells are constantly active, managing thousands of processes at once. One of the ways they stay organized is through the use of vesicles. These small membrane-bound sacs act as the cell's internal delivery system, that is transporting proteins, lipids, and waste with accuracy and efficiency. Now let's see what are vesicles. Vesicles are bubble-like structures enclosed by lipid bilayers. They are found in nearly all eukaryotic cells and come in different types depending on their function. Some vesicles carry proteins to the cell membrane for secretion. Others break down waste, and some store important chemicals. Common types include transport vesicles, lysosomes, secretory vesicles, and synaptic vesicles. In the illustrations given below, you can see the types of vesicles. Towards the left, you can see the transport vesicle, which transports the proteins to the cell membrane. And then the secretory vesicles, which carries the secretory proteins, and then through membrane fusion it releases the secretory proteins. And then in the middle you can see the phagocytosis and pinocytosis. Phagocytosis is the one in which the solid particle is taken in by forming a phagosome, and the pinocytosis is the one that involves the fluid particles. And finally, you can see the synaptic vesicle carrying the neurotransmitters from the nerve cell, which is released into the synapse formed between the two neighboring nerve cells. And then it is transported into the next one, thus the signal processing takes place in nerve cells. Now let's see the endoplasmic reticulum and vesicle budding. Vesicle formation often begins at the endoplasmic reticulum, that is especially rough endoplasmic reticulum. Here, proteins are synthesized by ribosomes and inserted into the ER membrane or lumen. These proteins are then packaged into budding vesicles, which pinch off and begin their journey to the next cellular station, that is Golgi apparatus. In the illustration, you can see the proteins from the endoplasmic reticulum being carried into the transport vesicle to the Golgi apparatus. Now let's see Golgi apparatus processing. Once vesicles reach the Golgi apparatus, they fuse with its receiving side, that is the cis phase. Inside the Golgi, proteins are modified, for example, by attaching sugar or phosphate groups. These chemical changes are essential for protein function. After processing, proteins are sorted and packed into new vesicles at the trans phase of the Golgi, which is ready for delivery to the next destination. So in the illustration, you can see the Golgi apparatus with the cis space, which is receiving the incoming transport vesicle. And opposite to that is the trans phase, where you can see the newly forming vesicle, and then the secretory vesicle, and then the secretory vesicle. Now let's see the types of vesicle transfer. Vesicle transfer can be intracellular or extracellular. Within the cell, vesicles move between organelles like the endoplasmic reticulum, Golgi, and lysosomes. In extracellular transfer, the vesicles carry materials to the plasma membrane and release them outside the cell. This includes processes like hormone secretion and neurotransmitter release. In the illustration, you can see different types of vesicle transfer. You can see the exosomes, which is transferred through exocytosis from the cell to the exterior of the cell. And then next you can see the endosome, in which the particles enter the cell and through vesicle trafficking they reach the Golgi apparatus and ER accordingly. And then you can also see phagocytosis and pinocytosis, in which the um, particles enter the cell. And then finally, you can see the exocytosis, which releases the proteins. You can see before that, you can see the proteins being processed from endoplasmic reticulum and then reaching the Golgi apparatus. And then uh, through exocytosis, they are transferred through the plasma membrane to the exterior of the cell. Now let's see exocytosis, that is releasing of materials. Exocytosis is the process by which vesicles merge with the plasma membrane and release their contents outside the cell. This is how neurons communicate and how hormones are distributed and how cells dispose of the unnecessary materials. It's a tightly regulated process essential for cell communication and waste removal. In the illustration, you can see the vesicle fusing with the cell membrane, and then the waste is released outside the cell. Now let's see endocytosis, that is material intake. In contrast to exocytosis, the endocytosis is the process by which cells take in substances. The plasma membrane folds inward, engulfing external materials and pinches off to form an internal vesicle. There are different types of endocytosis, that is phagocytosis, which engulfs large particles, and pinocytosis, which brings in fluids and solutes. In the illustration, you can see the types of endocytosis. The phagocytosis, in which a solid particle is engulfed and it forms a phagosome. And in pinocytosis, you can see the small solutes or fluid substances being engulfed and forming a vesicle. Now let's see the importance of vesicle transport. Vesicle transport is essential to maintaining cellular order. It ensures that proteins reach their correct destinations and lipids are distributed efficiently and the waste products are removed promptly. Without vesicle trafficking, the cell would lose its structure and function. This internal delivery system keeps cellular processes coordinated and efficient. In the illustration, you can see the vesicular transport overview, which involves the endoplasmic reticulum and Golgi and lysosome. You can see the ER from which the lipids and proteins are transported through transport vesicle to the Golgi apparatus, and then the Golgi modifies and it secretes, and then it secretes the vesicle, which is later transported either to the lysosome or to the plasma membrane, where it is released outside the cell. So this is the secretory pathway, and the other is endocytic pathway, in which the cells ingest the extracellular materials and they form endosome, which is early endosome, and then it forms late endosome, which then fuses with the lysosome, and the lysosome degrades the particles. Now let's see the disorders linked to vesicle transport. When vesicle formation or transfer goes wrong, serious consequences can arise. Defective vesicle trafficking proteins are linked to various genetic disorders, including Chediak-Higashi syndrome, where lysosomal trafficking is impaired, and neurodegenerative diseases like Alzheimer's and Parkinson's have also been associated with defects in vesicle transport within the neurons. So, vesicles are small but powerful transporters in the cell, from protein modification and secretion to the waste breakdown and even cell communication. Vesicles are at the heart of cellular logistics. Understanding how vesicles transport works helps us explore the fundamental biology of the cell, and also to uncover the causes of various diseases. Thank you for watching. See you in the next video.